With that said, I just, once again, just want to emphasize how fortunate we are to have had the support, and how important it is. And they, the American Lung Association made a commitment to fund research in lung cancer. They jumped right in with both feet. Not only are they funding the research, but what has been amazing for me is I've had more opportunities like this in collaboration with this organization to really educate people and bring research to people so people can understand kind of what we're doing and start to understand that there's a significant passion to bring discovery to each person. So I just, once again, I hope that as you hear Margie next, you hear Hoss, as you think of questions, as you hear, when you hear we're raising resources, we're advocating for research, it's faces like mine, it's my team, it's faces like Margie, it's Hoss, and it's that collective entity such as Fox Chase that are committed with organizations to make it happen. So that was kind of my big message, is I just want to emphasize that. The research, I love my research, don't get me wrong, but the opportunity here is to realize that we're as a team.
Thank you, Dr. Whetstine, for that important inspirational overview about research, and especially thanks for all that you're doing to change lung cancer care for patients. LungForce was not only created to support lung cancer research, like we just heard from Dr. Whetstine, but was also started to raise awareness that the mortality rate for lung cancer is increasing in women and decreasing in men. The American Lung Association also took on the challenge to end the stigma around lung cancer. Lung cancer can be a diagnosis for anyone, not just those who smoke. Dr. Clapper has been working since 1991 to pinpoint ways to find lung cancer in its early stages, when it is often more treatable and even curable. Dr. Clapper created one of the first basic research programs nationally to focus on developing strategies to reduce cancer risk among high-risk individuals and promote early detection and prevention. At the time, the idea of preventing a disease like cancer was not widely accepted. Today, she studies what is causing non-smoking women to contract lung cancer. It is now my pleasure to introduce Dr. Margie Clapper, Deputy Scientific Director at Fox Chase Cancer Center.
Well, thank you, Carolyn, for that kind introduction and for this opportunity. And good evening, everyone, and thanks for joining us. As Carolyn mentioned, the focus of my research here at Fox Chase is to identify who is at highest risk of developing cancer. And our hope is to develop therapeutic strategies that we can use to intervene in that process. Quite a challenge. We want to intervene as early as possible before this tumor even has a chance to begin to develop. And in my lab, we do that using a variety of different approaches. We use animal models, we use cell culture systems, and most importantly, in the end, human tissues. I want to share with you tonight our recent findings, which I think are most exciting. As Carolyn mentioned, we've been looking at never smoking lung cancer. This is a tumor type that I think is very understudied and one that needs a lot more research. And clearly a tumor type that is much different from what you would find in a smoker. There are many features that are quite distinct. Excuse me. You may not know that lung cancer is the 7th leading lung cancer among never smokers. So non-small cell lung cancer among never smokers is the 7th leading cause of cancer and death worldwide. And in the United States, as many as about 20% of lung cancers are diagnosed in individuals who have never smoked. Most intriguing and disturbing, actually, is the fact that the majority of these are in women, and we don't know the reason why. And it's this gender bias that started us 20 years ago to think about what might be the basis for this increased risk of lung cancer among never smoking women. And clearly, these numbers continue to rise. We asked ourselves, is it possible that something as obvious as the female hormone estrogen might be playing a role in this process? So my lab team is the first to discover that the human lung can actually metabolize estrogen. So it can take the female hormone and it can convert it to a number of derivatives. And most concerning is that some of these derivatives are thought to be cancer causing. In particular, one that's called 4-hydroxyestrogen. We've been able to also demonstrate that this process occurs in the normal lung and to a much greater extent in the lung tumor. And to us, this suggests that this process is probably contributing in some way to tumor formation in never smokers. With my colleagues at Fox Chase, we've established a way that we can actually measure these metabolites. And most importantly, we can measure the 4-hydroxyestrogen, or what we think is a carcinogenic estrogen. I call it the bad estrogen. And using this assay, what we've been able to do is to show that Asian women, unfortunately, are much more, they produce this bad estrogen better, so they're higher producers of this potentially cancer-causing estrogen. This finding is of significance because we know that the percentage of never smokers who develop lung cancer is much higher in the Asian countries, and particularly Southeast Asia, as compared to the United States. So as many as 60 to 80% of lung cancer cases in Southeast Asia are diagnosed in never smokers, as compared to about 20, as I mentioned, in the United States. We are now extending these findings, and we're partnering with the thoracic oncologists here at Fox Chase, in particular, Dr. Joseph Treat and Dr. Nick Boder, who have recently opened a clinic for never smokers with lung cancer to try and address the unique needs of this particular population. Together, using our assay, we have identified a group of never smokers who indeed are overproducers of this, what I call the bad, or we think cancer-causing estrogen. There's a lot more work to be done. We are now looking to profile additional cancer patients who are never smokers. We're looking in the community for healthy volunteers to try and figure out what is normal and what is abnormal in terms of these levels. And most importantly, we're trying to figure out how these estrogens might be causing damage within the human lung. It's our hope to be able to identify a high-risk population, a population that overproduces these estrogens, and then develop therapies that can benefit these individuals to try and inhibit or reverse that process, or even more closely monitor them for tumor formation. So I hope I have another opportunity to update you with our progress in the future. But for now, thanks so much for your attention.
Dr. Clapper, thank you for all your relentless work to help identify lung cancer in the early stages. You truly are saving lives. Now it's my pleasure to introduce Dr. Hossein Borghaei, who will share how research being done at Fox Chase Cancer Center in Philadelphia helps treat lung cancer patients, and what discoveries being made today from research means for the future of lung cancer patients. Dr. Borghaei is the professor of oncology at Fox Chase Cancer Center.
So hello, everyone, and thanks, Caroline, for that introduction, and thank you and your team for putting this together. You know, I really have to echo a lot of what my colleagues have said already on this program here. So I'm a clinician in the group, and my primary focus is to develop new drugs for patients who are diagnosed with various forms of lung cancer. I do have a small laboratory, but my primary goal has always been bringing new drugs into the clinical practice. And I guess a simpler way of sort of describing why it is important to have this kind of research and how to translate it into the clinic is the whole idea of immunotherapy that now has become a mainstay of treatment for many of our patients with non-small cell lung cancer. Why would I use that as an example? Is because really the research into how the immune system interacts with cancer dates back to many decades ago. If it wasn't for the work of two scientists who just last year received the Nobel Prize for discovering two separate pathways that has to do with the activation or the way the immune system interacts with the tumor, we wouldn't have been able to develop drugs that now we use as a matter of standard of care. So the research actually started 30 years ago, took a long time for us to figure out what we were doing. Drugs were developed. We took those drugs that were developed by scientists like Dr. Whetstine and Dr. Clapper, and we applied it to a patient population who was in need of better treatment for their lung cancer. And before you know it, we have so many different drugs that now interact with the immune system, and there are some wonderful results that we're seeing on the clinical side that it just encourages you to go and seek out these newer pathways, these newer molecules, these newer ways of addressing the cancer. So the kind of work that you just heard happening on the bench side, the kind of work that many of you help support by your donations and work for the organizations that we have just discussed, helps us develop these new drugs for patients who are in need of having drugs. Another example is the patient population that Dr. Clapper was just talking about. Never smoker patients. These are young patients. The youngest patient I've seen in my clinic was 26 years old. She came home from her honeymoon with metastatic non-small cell lung cancer. It just boggles your mind. Why is a 26-year-old never smoker getting metastatic non-small cell lung cancer? It just should not happen. So the kind of work that Margie described and the kind of work that Johnathan described can actually help us figure out why it is that these cancers develop and how can we better treat these cancers. We know a lot of these never smoker young patients who come to our clinics have specific mutations. The interaction between these mutations and the rest of the body is important. Finding targeted therapies are important, and also the ultimate goal of finding a cure for these cancers is what's important. And this is where all of you can participate and help us not only find patients who are in need of these treatments, but also how to help and support the research that's ongoing to come up with newer and better treatments. And as one of the NCI designated comprehensive cancer centers in the country, it is our job and it is our responsibility to bring you this kind of research and also to update you as to what we are doing and to promise you that these discoveries eventually will make it to the clinic and hopefully will lead to better outcomes for everyone who's in need of these treatments. So I think I'm going to stop there and opening up hopefully to your questions. Thanks.
Thank you, Dr. Borghaei, for your commitment in providing the best care possible for those battling with lung cancer. I'm now going to address the questions we received prior to the event, and if we have time, we'll open it up to those sent to Felice Kellum privately through the chat. The first question, I believe you all can talk on, it's give us an example of scientists and clinicians working together to find a better treatment for lung cancer.
Well, I can start with an example. So I've been here at Fox Chase for about 18, 19 years now. Throughout the years, working with colleagues on the science side, we've been able to identify specific, what we call pathways or molecular mechanisms where we felt that having available drugs allowed us to take the ideas from a laboratory and actually make a clinical trial around it and take it to the clinic and apply it to our patient population. So we've done that now, again, as I said, over the last 18, 19 years, several times. Unfortunately, they're not all successful in the clinic, but that's part of the process. It is hard to, promise clinical activity when you do these kinds of studies. A lot of things might look promising in the lab. We have ways of identifying what are the most prominent, the most promising, and then try to tailor those into clinical trials where we can actually check the hypothesis whether what we see in the laboratory translates into the clinic. I think we have another example right now. We're trying to put together projects with Dr. Whetstine, and I think Dr. Clapper mentioned her collaborations with a couple of thoracic oncologists here, specifically for the non-smoking patient populations. So perhaps, Johnathan, you want to comment about what you're doing with some of the groups here?
Yeah, so basically you make a discovery, and often the gap is being able to find your clinical colleague. The one thing that, recently I joined Fox Chase about just a little over a year ago, just a year ago, my lab moved here. And it has been an amazing experience because we're right next to each other. Our offices are just right next to each other. We can interact. And a recent discovery that actually was funded by American Lung Association's Lung Cancer Discovery was work on EGFR. It's a key oncogene that plays a key role in lung cancer. And we've made a discovery about how this is being modulated and controlled. And so we've met up with Hoss and discussed opportunities from what we found by looking at patients carrying this type of alteration to describe or discuss, can we now use our knowledge to potentially develop a marker, a biomarker to follow and have predictability about those patients that have that particular set of changes? Are they going to respond worse or better to certain therapies? And then more importantly, we're working with colleagues here to also develop molecules using chemistry because of the biology we've identified. So then we can put those into models that, say, for example, Hoss uses as well as Margie uses. So it's an active process. And that's something that's really important for, I think, a lot of people to realize is it's not that you can't bring the most fundamental, like basic discovery quicker to the clinic. It's how you and who you're placed around. And in this particular situation, you know, for us, we're very fortunate. We have colleagues who we can join forces with very quickly, take something that's early days discovery, but move it that much faster so we can partner then with potential novel therapeutics with whether it be industry partners or novel molecules that can be internally developed or other collaborators throughout the world. So that's just my example. It's been something that's happened, and Hoss has been amazing because he gives us as a scientist, me and my team, more of a clinical appreciation of what we need to be asking so that we ask the question even better as a scientist to help the individual.
Thank you. Dr. Borghaei, can you discuss benefits for stage 4 patients who have remained stable having radiation to lung tumor as long as no other meds exist, especially those with genetic mutations?
Yeah, so I think what the question is asking is this process of using what we call very focal radiation, stereotactic radiation, to small areas that are considered to be the primary source of the cancer cell. So in patients that have these molecularly driven tumors, which again we see primarily in patients who are never smokers, what the thoracic world is considering is the following. When we start somebody on a targeted therapy, we generally get very, very good deep responses. And by response, I mean we get actual shrinkage of some of the tumors, lymph nodes, and things like that. But it is rare for us to have someone with what we would describe as having a complete response, meaning complete normalization of radiographic imaging, all the disease going away. That doesn't happen a lot in lung cancer, and there are many different reasons for it. The field is moving towards an area where we are beginning to consider radiating specific sites of disease that might be escaping control, meaning that in a biological phenomena that we don't quite understand, we see patients where most of the disease is under control, not growing, but in 2-3 areas, all of a sudden the tumors start to grow. Because our impression is that the medication, these targeted therapies, these pills by and large, that are usually very well tolerated, are controlling the disease everywhere else but 2 or 3 areas, we go ahead and try to stereotactically, with minimal toxicity, treat these growing areas. What that does is that the radiation effectively kills the clone of cells that's growing in that 1 or 2 sites and allows the medication to do the rest of the work and continue to keep the disease under control. We've taken that and we are asking the question, now, what if we don't wait for these areas to grow? Meaning we get maximal response from the treatment, the disease is under control, we can still see some areas. Can we go ahead and radiate these to completely eliminate the disease everywhere we see it? That's a concept that's under investigation. It is not, as far as I'm concerned, the standard of care, and it has a couple of issues with it. The main issue for me is that we don't really know if the cancer cells in the areas on the CAT scan that we see are still actively growing, or is it just a scar or something else that we are observing. By and large, in patients that have these particular subtypes, these molecularly driven tumors, we don't think our drugs are killing the cancer cells. We think they're suppressing the growth. So the idea of offering some radiation to these areas that have now stabilized is an attractive one, because then you can potentially kill the cancer cells where we see them, but does not address this issue of what we call micrometastases, or small cancer cells that could be floating around. So I would say that it is something to be considered. If I had the choice, I would do it in a clinical trial setting, so we can learn from this experience, make sure we're not causing more harm and more damage. But it is an interesting phenomena that, again, is being intensely investigated. I hope that answers the question that was submitted.
Thank you. Dr. Clapper, has there been any advancement in never smoker lung cancer in the past year? What can we do to help ourselves to avoid never smoker lung cancer? Or is it just the hand we are dealt?
It's a great question. I think what we're learning is that immunotherapy is not effective, as sort of an extension of what Haas was discussing. We're finding that immunotherapy is not effective in never smokers with lung cancer, especially in those that have these driver mutations like EGFR. So I think what we're learning is that there's some suggestion that if we combine the immunotherapy with chemotherapy and use them in combination, that we might be able to sensitize these tumors in never smokers. In fact, we've started a similar trial here at Fox Chase recently that has been taken up by many institutions at the national level. In terms of what we can do to protect ourselves, that's a difficult one, because we don't know yet what the cause of this disease is. We can certainly avoid obvious things like radon and passive smoke, and maybe someday I'll tell you to avoid estrogens, although we know they do have some beneficial aspects. So beyond that, we don't know what's causing these tumors.
Thank you. So the next question is for Dr. Borghaei. I am an ALK positive and have been on elective for 24 months. Progress on the second generation TKI. Is there another third generation TKI in the pipeline aside from lorlatinib?
So there are multiple drugs for ALK-driven tumors that now are either FDA approved or on their way to become FDA approved. And this is a question that, again, requires a clinical trial to address it appropriately. And there is an NCI-sponsored clinical trials that we have available here at Fox Chase, trying to specifically address some of these questions. The short answer is that yes, there probably is one. I would say probably because we are now at a stage where once there is lack of efficacy with one specific drug, and there is evidence that the cancer is progressing, we tend to biopsy these sites and redo the mutational analysis on the cancer that now is growing and not responding to a particular drug. By doing that process, we've learned that we can actually identify which one of the other drugs that we have available to us could potentially be effective. So again, this is another one of those areas where there's a lot of interest in finding what exactly would be the best drug to follow after two ALK-driven drugs have stopped working. But it's not one of those things that I can simply say, yes, go on and take the next drug. It requires a little bit of an investigation and a little bit of work, which again, we'd rather do in a context of a clinical trial to make it a little bit more meaningful for everyone. But yes, there are potentially other drugs that can be used in this setting.
Thank you. Dr. Whetstine, this is a threefold question. Can you expand on your discovery and how DNA is controlled and impacts drug response? Are the changes you observe mutations? And is there a way to control the changes you observe?
Yeah, so the area that we work on is called epigenetics, and that's basically, if you think about it, epi meaning around, genetic meaning DNA. So it's that environment around the DNA and how it controls it. And what we actually found is that as you change that environment or how you control it, it allows genes that are very helpful to cancer cells, growth factors or oncogenes, genes that promote cancer, to be amplified, to be made more. By making more of those, they help the cell have the benefits that give them the growth advantage or the ability to resist certain therapies. And so what we've found is that this process of regulating it allows this to happen. They're not genetic events. In other words, we're making more of the DNA, but we're finding that it's not integrated or kept. It's not like your constant amount of DNA in the cell. It's made when it's needed, how it's needed. And the reason that's a challenge is that it gives the cell the ability to adapt to circumstance. If certain stresses or certain pressures are placed on it, it uses a program or it mutates genes, those genes that help drive that event. So those events then give the cell some plasticity. It can allow it to move around or respond differently, to adapt. So many of the things that we're looking at are not, quote, mutations per se. We're looking at how the cell is using the DNA based on a natural program that the average cell in the body has to help bypass stress. You think of, cancer cells and how they grow. It's a very stressful environment. If they're challenged with therapeutics, it's a very stressful condition. So basically, it's using this mechanism, how it regulates that environment around the DNA to control that.
Thank you. The next question is for Dr. Borghaei. How does research funding help in advancing patient care?
It helps tremendously. This is a question that I think I'm going to draw in my colleagues with Margie and Johnathan into it. Again, going back to the examples that we've used, immunotherapy. If there wasn't research funding, there would not have been discoveries made in the field of immunotherapy. Without those discoveries, we wouldn't have had all the drugs that we have now. And without those drugs, we wouldn't have been talking about potentially 10-15% of metastatic lung cancer patients being hopefully cured or having at least very prolonged responses without evidence of progression. So it directly relates to patient care. I think that the message or the concept that I think we have to talk about is that when a scientist talks about doing something on a tissue culture, on a plastic, or when you hear about an investigator saying that a certain mouse experiment showed promising results, these are actually potentially sentinel events that can have significant implications down the road for patient care. It does take a long time to go from a laboratory finding supported by direct research funding from the NCI, the NIH, or philanthropy, or organizations such as yours, to developing a new treatment. That is a bias, and that's something that's always been there, and it will always be there, because science is precise and it's as accurate, and it has to be done in a way that leaves little room for doubt, especially the kind of science that you want to translate to the clinic. But without what Margie does and without what Johnathan does, people like me would use drugs that were made 40 years ago, and none of us are happy with drugs that we developed 40 years ago. That's why we have all of this, so that we can find better treatment, so that we can answer the kind of questions that you heard from Margie or the kind of questions that you heard from Johnathan. So to me, and you can call me biased because I work with these wonderful people, but there's a direct correlation between what I hear from Margie and what I hear from Johnathan and what I take to the clinic directly to my patients, right? So I don't know, Margie, if you want to comment a little bit more on that.
Sure. My lab is set up uniquely, so we're mouse. My daughter calls me a mouse doctor. So we do a lot of mouse modeling and preclinical work, and our intent in everything that we do is to take that to the clinic and impact care in some way, and then use what we learn there to come back to the laboratory and ask new questions, whether it's in our mouse models or at the bench, whatever. So without having both components, the wheel doesn't turn. I think hopefully we're fueling the clinic, and the clinic is fueling us back with their discoveries and their observations. And I've been at Fox Chase a very long time, and that's what's really kept me here, is that that wheel does turn here, and everything we do in some way impacts the clinical setting. And Fox Chase is a wonderful environment to be able to take what we learn at the bench and quickly test that in the clinical setting, because we interact with the clinicians on a hourly and daily basis. So I think all of us at Fox Chase, the intent of everything we do is ultimately to bring that to the population that will benefit the most, whether it's in the community or it's in the clinic. And many of our ideas are high risk and out of the box, perhaps. So that's where the foundations are extremely important, because high risk ideas don't always go over very well for national funding. We have to make sure they're very concrete and well documented. So funding from organizations like the ALA is extremely important to get a new idea, an innovative idea, a high risk idea off the ground, so that we can then go seek funding from federal government and other sources.
I would like one other thing to point out is I was fortunate in 2004 to be on a team that discovered a gene that had a unique function. And people have been looking for it for a long time. That was in 2004 in December. Currently, that not only has the gene been expanded upon and it created this whole area that I was just telling you about, but that is actually in early stage clinical trials currently across the multiple cancer types. So it went from the discovery of a gene that regulated a process, understanding the mechanism in a lab, to now it's actually being done in a clinical setting. And, you know, as a more close to home example, is actually being pursued or thought about in placement for trials here at Fox Chase. So what people need to realize is that in that 15-year window, we went from not knowing anything about it. In fact, the field debated whether the chemistry could happen, whether the gene actually existed, to from one gene to now more than 20. And now there's multiple industries being built around it to develop novel therapeutics to target it. And there's actual clinical trials in place today from multiple industry partners that are pursuing this, both for pediatric malignancies, adult malignancies, solid malignancies such as lung cancer, as well as in blood cancer. So that is, without those original findings, the basic question, just the fundamental thing of what does a gene do and how does it work, we wouldn't have these trials today.
Thank you. So we do have a few more questions, but I wanted to see, Felice, have you been receiving any questions?
Not yet.
Okay. So we'll just keep going with the questions I have. I know we're 10 minutes over. I think we'll go to about 6:20. So I'll just keep going, and Felice, just interrupt if you get any questions from the group, okay?
Sure.
The next few questions are addressed to Dr. Clapper. Are women who take hormones at a higher risk of getting lung cancer?
So that's the million-dollar question. I can tell you what we know. So there was a very large study that was done years ago by, it's called the Women's Health Initiative. And what they found was that if women took estrogen in combination with progesterone, they were at about a 2-fold increased risk of dying from non-small cell lung cancer. Of course, this was very alarming. There's now 20 years of follow-up data, and it shows that eventually that risk will subside, attenuate, but still not back to what you would expect if those individuals had not been exposed. On the flip side, there are also studies that show if an individual takes an anti-estrogen, that might confer some protection. So there was a study that was done looking at breast cancer patients who took tamoxifen and then went on to develop lung cancer. And that particular study showed that if they had taken tamoxifen, their lifespan was longer after they developed lung cancer. So there is evidence that the estrogens are playing a role. Clearly, we have much more to do. In our studies, what we do is we collect hormonal data on each patient to try and correlate exposure, whether it's from supplements or through pregnancy, infertility medication, et cetera, and try and correlate that exposure with their estrogen profile, because we're very interested in trying to answer that question.
Thank you. And then one more, is there anything that can be done for someone who makes lots of bad estrogen?
So that's our ultimate goal, to intervene in that process. We don't have a therapy for that now. But what we're trying to do is identify either drugs or natural products that could stop the production of these bad estrogens, either reverse the process or somehow influence it. So we're working hard to try and identify what those agents are. We're not there yet, but we do know the enzyme that is responsible for producing that bad estrogen. So that's a start, and it's our target, and we continue to try and do that.
Great. And then actually one more is, does this process contribute to lung cancer in men also, or is it just women?
Okay, that's a good question too. So we have been able to profile the estrogen metabolites in both the lungs of men and women. And I can tell you this process also occurs in men. The difference between the men and the women, though, is that we see much higher levels of the metabolites as well as the parent estrogens in the female lung as compared to the male lung, as you might expect. But I do think in some way it's probably contributing to both.
Thank you. Hey, and if we don't have any questions, there's one more question for Dr.
I have a question. So the question is, how has COVID impacted Fox Chase's ability to conduct clinical trials?
I'll take that one. Fox Chase was in a unique position because of the fact that we are not what I would call a general hospital. We're only a cancer center. And through close collaboration with our colleagues at Temple University Hospital, Temple Health System, and Jeans Hospital, we managed to maintain Fox Chase as COVID-free as possible, meaning that if there was a patient suspected of having COVID, or if there were issues with someone who might have been exposed and they needed to be seen in a hospital setting, through these agreements, patients were diverted to other centers. So we were extremely fortunate that we had a system such as Temple Health System that agreed to work with us and to keep our cancer patient population hopefully not exposed to this devastating virus. So from a clinical trial perspective or clinical operation perspective, I have to tell you that patients who needed to get treatment in terms of chemotherapy, radiation, who absolutely couldn't be delayed, were able to receive their treatment. And by extension, if someone was an appropriate candidate for one of our trials, we were able to enroll patients on a study. Things were slower for sure. Many of our colleagues in the clinical trial office had to work remotely, so things took a little bit longer to do, but by and large, we were able to maintain a steady pace of accrual to clinical trials at Fox Chase, so we were not impacted as much as some of my colleagues in other centers where things actually had to shut down completely. Like, I can understand in New York area, the priorities were completely different. It wasn't so much clinical trial, but it was to avoid further devastation by this virus. So we were in a good position. Everything is again fully functional and has remained fully functional. So we did not get impacted by that as much.
Thank you. Elise, are there any more questions?
No, we're done with questions.
Okay, great. Well, I think it's a good time to wrap up. We're about 15 minutes over. We can send out a survey to everyone in the next 48 hours to kind of get feedback, since this was our first education session with Fox Chase. And I think there's a lot more information that we can share. But thank you to all who called in and participated. Thank you to Dr. Whetstine and Dr. Clapper and Dr. Borghaei for their time and their knowledge. So if you have any other questions, you can email felice.kellum@lung.org, and we'll make sure to get them answered by our panel of experts. And just a couple things, from June 14th to June 20th, the American Lung Association will be holding their annual turquoise takeover nationwide. Buildings will be illuminated turquoise. Lung Force walks will be taking place virtually, and we need as many people nationwide to help us by spreading awareness and knowledge with the color turquoise. And to register for our virtual Lung Force Walk Philadelphia, which our opening ceremony is going to be on June 20th at 10 a.m., you can go to lungforce.org/Philadelphia. You can also join Dr. Whetstine, Dr. Borghaei, and Dr. Clapper's Fox Chase team directly by visiting action.lung.org/go-to/FoxChase20. Thank you, everybody, so much. I hope you have a wonderful evening, and we look forward to seeing you again soon.
Thank you, everyone.
Thank you.